Could be real.
You, sir, are the man.
Well, they were glad to help you. So I'm glad it all got taken care of for you.
They looked up the Florida Department of Higher Education, fixing to call it on that college, because they even tried calling and saw that I would just get sent to a voicemail that didn't have a name or anything to it. It was like a all-tell voicemail, and they couldn't get nobody on the phone. And the other lady that you got on to for talking to me, she got real ill with them. And they was able to make some calls and get something done before they could see that I never really even went there. And made it where I don't have to have the transcript now.
Well, that's great.
Thank God. So then that issue arose and I didn't take exam 3 in between lecture and lab today. Do you want me to just stay after Monday and take it?
That'd be fine.
Okay.
Monday will be fine. So. Here.
Where is the writing department in here?
The,
Like writing?
probably over there in the next building, I think. Oops, that would be really fun. Come on. Okay, we're going to go ahead and get started here. We're going to finish up our discussion on the digestive system and something to do with the brain. But anybody have any questions on anything before we start? Okay. Then let's go into here, and I got us all the way down to we had just finished up looking at the consequences of acid reflux, gastroesophageal reflux disease. It occurs when stomach contents penetrate the cardiac sphincter, and it goes up, and the stomach contents are very acidic, and they will burn the duodenum. The duodenum does not have the tight junctions to hold the cells together. It doesn't have the alkaline mucus that the lining of the stomach does. And so when we release contents from the stomach up the esophagus, it causes burns, and the burns look like what we're seeing here. This inflamed tissue is burnt tissue, because that's, essentially hydrochloric acid with a pH of 2 hitting that unprotected tissue and causing the burns to occur. This is gastroesophageal reflux disease. And once we start treating it, it's practically impossible to stop treating it, because once it starts, it will often continue. Now, a lot of times it is triggered by what we call the hiatal hernia, where we tear a hole in our diaphragm and a portion of the stomach protrudes through here.
I had that happen to me.
Okay.
I was 18 and they said I had an ulcer that was like the size of my, like the entire lining of my stomach was
Mm-hmm.
an ulcer. And I went to do something one day and all of a sudden this knot popped out right
Mhm.
between my ribs and it hurt so bad.
Oh, yeah,
I had to, like,
yeah.
push it back.
Yeah, that's a very common thing. As I think I said this last week, that about 80% of the population has a hiatal hernia at any given time. So it looks something like this. We get a bulge of the stomach up through the tear in the diaphragm. So, okay. On that bright note, we'll move on to more interesting stuff. Now, the small intestine is about 20 feet long. The small intestine is about the size of your finger, and this is where most of our absorption is going to take place. It goes from the pyloric sphincter to the ileocecal valve, and has 3 parts to it. It has the duodenum, the jejunum, and the ileum. Now, there's a model of the small intestine here, which is highly detailed and very specific. That was sarcasm. But it is about 20 feet long, and it's tucked into the area in the abdominal cavity. It's wrapped around on 3 sides by the large intestine. And so there's the front of the small intestine. Can't see much, here. The jejunum is the large middle segment. The duodenum is the first 10 inches or so. And then we have the ileum that comes after that. And the ileum is where where the structure that connects to the large intestine. And
That's the center area.
Hmm?
You said that. That's
yeah,
just the name of the
it's
center area?
it's from about a foot after the start of the small intestine. If you go about 12 inches past the pyloric sphincter through the duodenum, the jejunum starts there, and it runs for another 10 or 12 feet or so. So the jejunum, the small intestine, is held in place by the mesentery structures we looked at last week. And it's covered by the visceral peritoneum with the serous fluid in between there, because these things are going to move. The structures are going to move as we process food through them. Okay, now we flip it over on the posterior surface. We can see a couple of we can see 2 valves here, or we can see where the pyloric sphincter is. There's the first 10 inches. There's the duodenum. This is where the pyloric sphincter empties into the small intestine right here. Over here is the ileum, and that little structure there is the ileocecal valve, where the small intestine and the large intestine connect. Now, the duodenum starts right at the ileocecal. It starts right at the pyloric sphincter. Remember, the pyloric sphincter is the sphincter at the end. Let's figure out. There we go. That's the right angle. The pyloric sphincter is the end of the stomach, where it empties into the small intestine. The pyloric sphincter is located right here. And this is the first 10 inches of the small intestine. I'm trying to get it so it shows the way it should. So this is the duodenum. You notice these ridges in here. The pancreatic duct. The pancreatic duct is this structure that runs into the duodenum and releases pancreatic enzymes to break down protein. The green structure is the bile duct coming from the gallbladder up here. Now the gallbladder is receiving bile from the liver. It stores it in the gallbladder and is released through the bile duct at the same time that the pancreatic duct is releasing the pancreatic juices. So bile breaks down fat. The pancreatic enzymes break down protein. That's all taking place in this first 10 inches or so. So we chop up fat here and we finish the breakdown of protein. The duodenum is very, very short. It's only about 10 inches in length. And then we get the jejunum and the ileum. Both of those are designed for absorption as opposed to breaking down the product. So while there's probably some absorption taking place in this first 10 inches, most of it's taking place in the jejunum and the ileum. So, and this is where the pancreas gets to show off its double duty function, because we know the pancreas releases insulin and glucagon. That's its endocrine function, but its exocrine function is to release pancreatic enzymes and our bile to chop up the fat. And all the bile does is take the fat and chop it up into little tiny pieces. And that's all, because that's how we can get it absorbed. So now, a better perspective of this, let me see if I can get this one to come on. Here we go. Here is, if we look down here, you can see on this structure, you can see the location of everything where it's supposed to be. Because here we see the liver right here. There's the gallbladder right there in green. And the gallbladder really is green. The presence of bile in the gallbladder gives it a greenish appearance. And here we see the stomach here. This is the pyloric sphincter here. And you just see the beginnings of the duodenum right there. And you can also see the pancreatic duct here leading into the duodenum. So this gives you an overall perspective of where things are here. So last week we looked at the esophagus and saw where the esophagus is located. Here, there's the cardiac sphincter there. And then now we're dealing with the small intestine down here. And if we look a little further down, you can see how the large intestine wraps itself around the small intestine here. So held together by the mesentery, if you will. Okay. Anyways, this is what the backside essentially of the small intestine looks like. And this is pretty much life size. Let me get the model of the small intestine. I want to get a good perspective on just how big this is or how large this is. Let's look at, switch cameras. Okay. Holding this in my hand, this is just how big the large intestine and small intestine. It's slightly smaller than 1:1. So this came out of the torso model. So the large intestine's about 6 feet long. If you stretch it out completely, the small intestine's about 20 feet long. It's no bigger around than your finger and curled around to get that 20 feet. The large intestine is, you can see how it stretches. If I stretched it out, it might be 6 or 8 feet in length, maybe. So again, not a very large structure. that we're dealing with. Okay, so now let's move on. The ileocecal valve is the connection where the ileum butts up against the small intestine. I'm sorry, the large intestine. There is a pouch on the large intestine called the cecum, and this valve sits right above the pouch itself, so the contents of the small intestine are being discharged into the large intestine, and it settles in that pouch first before it starts moving its way upward. So now, what does the small intestine look like in cross section? Well, like the rest of the digestive system, we have two layers of smooth muscle, longitudinal and circular. The outer layer is the serous membrane, where it has, with the visceral peritoneum and the parietal peritoneum above that with the mucus in between. We have below the serous layer, we have the two layers of smooth muscle. Below that, we have the submucosa and the mucus layers. And here we are open to the inside. Now this is the lumen down below here of the small intestine, and you can see that these are all simple columnar cells. The simple columnar cells are designed to allow content to pass through. So we absorb. And behind the simple columnar cells are capillaries, fenestrated capillaries, just like the same type of capillaries we have in the glomerulus, except instead of filtering, it's for absorbing. And so if you look, every one of these are simple columnar cells, and they're all lined up. And if you look carefully, you can even imagine that you can see the goblet cells, like there's one, there's one, there's one. And these darker spots are all the goblet cells, because this is covered with mucus. So let's put the labels on and see if I got it right. Serous membrane, smooth muscle, both longitudinal and circular. The submucosal layer, which is the basement layer for the mucous membrane, which is what we find here. And then we have the lumen of the small intestine. And we increase the surface area of the small intestine tremendously by having folds in here. We have villi. Let's take it. Villi means fingers. So we have projections on a small intestine called villi to increase the surface area. We have microvilli, which are little fingers that come off the fingers to increase the surface area. And we have rings of tissue in the small intestine itself. So the total surface area of the small intestine is probably going to cover the floor space of this room. It's a huge amount of surface area. Because if we were to take it, stretch it out flat, it would cover everything. So, and but this is essentially where we're going to be absorbing our nutrients from. So when do we get stuff into the small intestine? Well, about 6 hours after we eat. It passes through the pyloric sphincter. We don't get it in there right away, but it gets through the pyloric sphincter. But in the duodenum, we still have to break down the fats. We have to break down the proteins. We still have to finish any carbohydrate digestion, which didn't get stopped. Well, it should have gotten finished in the stomach, but if it didn't, we have another chance for it here. Now, the pancreas and the liver play a big role here. The pancreas is where we release our digestive enzymes on the proteins. The liver releases bile. The bile, is there to chop up the fats. Bile acts like a detergent to break up the fats into small pieces. Anybody that tells you that they can't eat fat because they have no gallbladder is mistaken.
It just runs through, right?
Yeah. So, because the liver is still going to make bile. You may not be able to eat a whole pizza or a whole cheesecake, but you can still eat fats. We still have to have fats in our diet. Now, a couple places, most of the absorption takes place in the small intestine. The duodenum generally picks up iron. I don't know why iron is selected to come out in the duodenum. B12 and the bile that went through get absorbed at the very end of the ileum again. Water and fats are picked up throughout. So we are going to absorb nutrients all the way through here, except for these, but there are these exceptions. So the duodenum, the jejunum, and the ileum. First part, the duodenum, 10 inches long. The bile duct and the pancreatic duct come together in here and release bile and release these digestive enzymes. There is a sphincter here called the hepatopancreatic sphincter that controls the release of the bile and the pancreatic juices together. Now, the reason I like talking about that is well, first of all, there's the duodenum right there. There's the pyloric sphincter. Now, these ridges in here in the duodenum are called the plicae circulares. They're just ridges in here. And the ridges are the initial effort to increase surface area. We increase the surface area. Now we have the villi, the fingers coming off of the ridges, and we have the microvilli, the little fingers coming off of the fingers. That's how we get this huge surface area. The ability, there's the going off to the jejunum there. Okay, yeah. So the plicae circulares are going to have villi and microvilli to increase the surface area for absorption. We're going to see that pattern all the way through the small intestine. And it looks like this. Plicae circulares. The reality is, in the picture on the left, you can see the rings here, the folds, the mixture, the chyme that comes out of the pyloric sphincter actually spirals around. You know, it sort of looks like the inside of a vacuum cleaner hose, and it spirals through here as it's being sucked down. You know, and we keep mixing it with bile, and we keep mixing it with these enzymes to break down the proteins. And the folds have capillaries, they have arteries and arterioles and venules, and they have lacteals. Lacteals are part of the lymphatic system that allow us to absorb the fats, because the fats don't go into the capillaries. They go into their own type of capillary called a lacteal. Now, eventually the fat's going to end up in the blood, because all the lymphatic system ends up going into the blood anyway. But we absorb the fat through our own, through the lymphatic system itself. We can't see it on here, so but here is, we can see the lacteal here. This is a villi. This is a finger projecting off of the plicae circulares. So this is a finger projecting off of that, and then coming off of the villi are the microvilli here, these tiny projections out here. So we have the rings with villi and microvilli coming off of them. We have the capillaries back here to pick up nutrients that are absorbed through the simple columnar cells. Those capillaries are fenestrated capillaries. They have holes in them. So instead of filtering, they're going to be absorbing. And then in the middle of all that, we have the lacteal. The lacteal is part of the lymphatic system. The lymphatic system is the parallel circulatory system. I was talking about fluids and electrolytes in lecture today, and I was talking about how plasma becomes interstitial fluid. Interstitial fluid is collected as lymph and brought back to the plasma. We have the lymphatic system has its own circulatory system of vessels that collect this lymph, this saltwater fluid. But when they're in the small intestine, we use them to pick up fat, chopped up pieces of fat by the bile, gets absorbed by the lacteals, and then brought into the plasma that way. Because all the lymphatic vessels from all over the body, bring the interstitial fluid back, run it through lymph nodes, and then dump out at two large valves or ducts at the subclavian veins up here. And then it's back in the blood. And that's how we get the fat into our blood. So, and of course, then the fat all goes to the liver, and we end up using the HDLs and the LDLs to distribute the cholesterol around. So, okay. Now the pancreas, we've talked a lot about the pancreas. Pancreas sits just below the stomach. It is we talked a lot about its endocrine properties with its islets of Langerhans and the alpha cells and beta cells that make glucagon and insulin respectively. But the exocrine functions involve producing pancreatic enzymes to break down proteins. The acini cells, the cells that surround the islets of Langerhans. The islets of Langerhans sit in their little clusters, and we make glucagon and insulin out of them. But all the cells around them are the acini cells, and they secrete the digestive enzymes. Now, to protect the small intestine, the pancreas also produces an alkaline goop solution, whatever, that comes out and coats the duodenum, because the chyme that's coming out of the stomach is very acidic. We don't want to burn the small intestine. I'm sure there's probably another, if gastroesophageal deals with acid reflux, I'm sure there's got to be another name for burning the small intestine too. I just haven't heard of it yet. But the acini cells secrete an alkaline solution to neutralize the chyme so that we don't burn anything. And now we release these pancreatic enzymes, pancreatic amylase to pick up any carbs that got through, lipase to break down any fats that the bile didn't get to, nucleases to help break down additional proteins, as well as proteases. So the acini cells are going to be busy releasing nothing but enzymes to break down, do the last bit of digestion there in the small intestine. So, and there's our duct. The cells of the duct are going to be releasing the alkaline solution to neutralize the small intestine, neutralize the chyme. The acini cells are located all around the islets of Langerhans. So again, spleen, pancreas, duodenum, the tail of the pancreas. There's a hard-hitting factoid for you. In the body and the head, the main pancreatic duct, the accessory pancreatic duct. So a lot of times, if you're trying to identify a structure and it's similar to the main structure, it's often called the accessory. So it just sort of, anatomists weren't very clever when they were naming things. So, okay. So this is what the acini cells look like. This is an islet of Langerhans right here in the light pink. The dark pink, where you have all these other cells, are the acini cells, and they're busy making the pancreatic juices, the pancreatic enzymes. And that's a lot. They make 1.5 liters of these digestive enzymes. So, and it's all coming down into the small intestine, into the duodenum. So now we get to the large intestine. Large intestine is about 5 feet long. It's called large because it is larger in diameter than the small intestine. The small intestine is about the size of your finger. The large intestine is about the size of a baseball. So the large intestine and the small intestine come together to ileocecal valve, and the valve controls the movement of any undigested material, because that's what we're receiving here. Material that wasn't digested, stuff that's made of fiber, stuff that we can't process, stuff that got through and was not broken down. And that's what's getting into the large intestine. So that comes through the ileocecal valve and dumps into the cecum itself. And then coming off the cecum, we have the appendix. So the large intestine, excuse me, is broken down to the cecum, the appendix, the colon, the rectum, and the anal canal. And so the cecum itself is right below the ileocecal valve. Let's get a picture up here. Here in a second. Now, what about the appendix? You know, you hear a lot of things about the appendix. Is the appendix, it's been called a vestigial organ, an organ that has no use anymore, no role. And that's not true. The appendix is used, well, first of all, it acts somewhat like our tonsils do in the sense that it will sacrifice itself to detect a bacterial invasion. You know, it will become inflamed and engorged and cause difficulty. Now, we don't know how often that happens. When it gets out of hand or becomes a significant inflammation, then we have appendicitis. Not every appendicitis case requires surgery. Many can be treated with antibiotics. But if it's near rupturing, then probably yes. But the appendix is designed to sacrifice itself to alert the immune system that something's going on. It also is often used to store healthy bacteria. Because when we take antibiotics, for example, when we take an antibiotic, we destroy all the bacteria in our colon, in our large intestine. Everything's killed. But we preserve part of our normal flora of bacteria in our appendix. And so we repopulate the flora in the large intestine this way, because we've killed everything off. Antibiotics give a clean sweep. They kill everything, or mostly everything. But the appendix, if we're storing bacteria in the appendix, it can repopulate with what we call our normal indigenous healthy bacteria that belongs to us. And if we have lots of our own bacteria covering a surface, pathogenic bacteria don't have a place to go. You know, we keep bacteria, we keep pathogenic bacteria away. There's no place for it to land. You know, it's sort of like if you wash your hands with antibacterial soap, you kill off everything and it stays dead for hours. There's no living thing on your skin. And when the soap wears off, there's nothing there to protect you from any pathogen finding a home on that open space. Whereas, alcohol kills everything for about 20 minutes. You know, a hand sanitizer kills off all the bacteria for 20 minutes, but then your own healthy stuff, you know, on your arms will repopulate that area and you'll be protected again. So not a big fan of antibacterial soaps. Hand sanitizers, yes, you know, because it's all about timing. You don't want to have a dead area on your skin for hours thanks to this, you know, antibacterial soap. So anyway, what we do here is, you know, a patient that takes a lot of antibiotics kills off their bacteria in their gut. We need that bacteria. We can repopulate from our appendix so that we don't have some sort of opportunistic pathogenic bacteria come in and latch onto a new location. And the appendix may have something to do with B cells and T cells in fighting off specific types of infections. So the appendix is not a vestigial organ, as it's often said, a useless organ. So here's where the appendix is located. And you can see here on the model, right here, there's the cecum. this is the cecum, this is the pouch. There's the ileocecal valve right there. And here's the appendix there. The ileocecal valve comes in here, opens up right there, and there's the appendix projecting off there. So, and I might get a better view on big model here. If I can hit this. There we are. And on this model, you can see there is the end of the ileum here, coming up to the ileocecal valve right here. And there's the colon. This is part of the ascending colon here. And this is the cecum, the pouch here. And then down here is the appendix. It is attached to the edge of the small intestine by some mesentery. But there's no connection to the small intestine. And that's our appendix, you know, and it has a very useful purpose for us after all. Let's get back to our, okay. So there's our large intestine diagram. You know, we see it looks very much like what our model looks like here. You can see the appendix, you can see the ileocecal valve, you can see the cecum. Everything that goes into the large intestine goes into the cecum first, and then is gradually brought upward and around and over through peristalsis. We go from the ascending colon to the transverse colon to go across, and we have a descending colon to come down. We go into the sigmoid colon, we lead into the rectum, and then the anal canal, and of course the anal sphincter at the anus. So, and we are processing this whole time to remove water, create vitamins. Vitamin K in particular is manufactured here in the colon by the presence of the bacteria acting on the indigestible stuff. This is where the fiber ends up. If you have bran flakes, if you have raisin bran, if you have anything with fiber, if you like beans, the fiber inside beans ends up in here because we can't process it. You know, it's just, we're not cows. We can't do anything with it. You know, we can enjoy a salad, but the indigestible fiber in the salad, the cellulose in the leaves ends up in the large intestine. Can't do a thing with it. So.
My aunt just had surgery, like she's still in the hospital, and had all but like an inch of her colon removed.
Mm-hmm.
Because like colon cancer runs in their family,
Mm-hmm.
and they couldn't even do a colonoscopy anymore because they couldn't get around the fin.
Mm-hmm.
But she's been really sick with diverticulitis for a long time.
Mm-hmm.
But isn't that from like a nut or something getting stuck in a fold and infected?
Oh yeah, that's exactly what it is. Diverticulitis is there are pouches on the inside. I'll show you here in a second, but there are pouches inside the large intestine called diverticuli, and you process food through there. If those pouches get inflamed, it's called diverticulitis, and it's caused by seeds, or corn kernels, or anything like that, peanuts, and it can be seeds as tiny as seeds in strawberries, because my mom had an attack of diverticulitis one time for eating strawberries. There's little tiny seeds in the strawberries, and who would have thought? Normally you think of something like sunflower seeds or peanuts or eating popcorn or something that's going to get packed up in there, but it can be extremely tiny seeds. It can be large seeds, and there's two views about diverticulitis. One says never eat anything with seeds again, and the other view says don't eat anything with small seeds like strawberries.
She told me that she got some cool pictures that she was going to send to me because she got them for me because she thought that was cool.
Oh, okay. And so you can, it is a very painful condition. The pain, my mom described it, the pain is like a heart attack, but it's on the lower left side, and you run a fever and you ache all over and you feel terrible. And as soon as the antibiotics start to work, you start feeling better right away. It's treatable by antibiotics.
They tried every antibiotic they could and it would not
Mhm.
clear up, and they finally said that they had to go in. They only planned on removing, I think, like 4 inches, and
Mhm.
then they ended up only leaving like the last inch right where it attaches to the rectum or anus.
Mm-hmm.
And she says that she feels better, she felt better as soon as she woke up,
Mm-hmm.
but she's still not much. I think she comes home this week sometime.
Well, that'd be good.
But
Yeah.
she was real sick. She lost like almost 100 pounds.
Wow.
Couldn't eat anything. Tomato
Mhm.
products made her really sick.
Mm-hmm. Because of the acidity, probably. Yeah. So, yeah, once it starts, it's becomes very challenging then to avoid, especially if you're afraid of running, having an attack again.
She was actually on a mashed potato diet.
Okay. Okay. Well, I like mashed potatoes, so. So, okay. So let's see what we got here now. On the large intestine, its primary role is to get water out of the waste. Now, the amount of water taken out is very important. If we take out too much water, we become constipated. If we take out too little water, we develop diarrhea. So we try to have about the fecal matter should be about 30% solid and about 70% water. It doesn't take much water either way, though, to give us problems. Lots of medications affect the ability to retain water or not retain water. Antihistamines, are going to shut down the release of water from the tear ducts and the nasal cavity, so your eyes don't water and your nose doesn't run. Because if you're allergic to something, well, that also may have an impact on your colon too, because you may not have enough water, being, you may be taking out too much water from your colon. Pain medications often work by, they block pain signals. Well, the morphine-based pain medication tends to slow down the movement of the large intestine. And so your patients taking lots of pain medication often become constipated because they affect the amount of water, that's left behind. So a lot of little things come into play here. So, okay, so the large intestine is going to produce the fecal matter. That's the end result of processing the waste material. The color of the fecal matter is based on the presence of pigments left over from breaking down hemoglobin. And so we can tell a lot about the color of the feces, depending on shades of brown or green or yellow. It tells us a lot of what your patient's been eating or how they've been processing food. If it's black, it might indicate that they have an internal bleed somewhere. So there are actually people that have studied this, believe it or not. I don't know how they kept a straight face the whole time, but that's a whole nother story. Anyway, the vitamins, yes, yeah, I know. Yeah. I can barely clean up after my dog, so how could, so vitamins are recovered here, particularly vitamin K. Vitamin K gets made here. Vitamin K is essential for clotting. We recover water, we recover electrolytes. That's the primary role here. We want to have feces that are about 70% water, and anything more than that, we want to have recovered. So, and it is larger in diameter, but much shorter. Now, a couple of things about the large intestine is that we lose the longitudinal layer of skeletal muscle. We keep the circular layer. The longitudinal muscle gets reduced to a long band that stretches across and around the large intestine. It's called the tinea coli. And because of the absence of the longitudinal muscle per se, we get the large intestine to pucker up into sacs. We call them hostras. And Then we have bags of fat that hang out off of the peritoneum too. We don't know what they do, but it looks like this. This is the Tinea coli here. This is the band of longitudinal muscle. These are the pouches that form. And within the pouches are smaller pouches, the diverticuli, that can become inflamed. And this yellow stuff here are fat that hangs down off of the off of the peritoneal wall. Let me show you on the model here, even though this is such a good picture. Okay, let's take a look at our model. Now, first of all, this is the Tinea coli. This is all that's left of the longitudinal muscle. These pouches here, those are the hofstra, and those are the little bags of fat that are hanging down. And it shows me nothing. Okay, so, well, I hadn't taken this apart for a while. I forgot what was underneath there, which is nothing. So anyway, let's move on. This is still a pretty good picture here, though, showing what's going on. And
What did you call that? The fat, that's the epilogue.
oh, the fat are yeah, they are the epiploic appendage. The epiploic appendage. Okay, and that's what we see there. We don't know why it hangs out like that, but it just does. You know, we'll probably discover in a couple of years it's very important for us or not, you know. So, but we definitely can see the band of longitudinal muscle here and the pouches there. So. Now inside the large intestine, it's mostly simple columnar. We've seen simple columnar all the way through the digestive system in a lot of places, pretty much all the small intestine, all the large intestine. The only place where we change this is inside the anal canal, where we have stratified squamous, because we're going to have wear and tear in the stratified squamous. You know, and we don't want that. You know, wear and tear is not good. We have stratified squamous in our mouth because we eat things. We have stratified squamous at the anus because things have to pass through there. So lots of goblet cells line the large intestine to lubricate. We have to get, to get the fecal matter out, we have to make sure that there is no pain involved here. We have to lubricate everything with these goblet cells. And of course, there's our large intestine right there. Look at all the goblet cells. All these little white things are goblet cells. So these are all simple columnar cells in through here and here. The one thing we do notice is that they are organized into structures known as crypts. Those are those little folds that form, that go deep down inside there. The crypt, for good or for bad, provides an opportunity for bacteria to grow. In this case, it's probably a good thing, because that's where our bacteria grow that are processing the waste material in the large intestine. In other parts of the body, the crypts are used to let bacteria grow that are harmful. Like we see the same type of crypt structure in the tonsil, for example, giving a place for the bacteria to grow. Here, it's probably a good thing. But you see, an extensive amount of goblet cells here lubricating everything. Okay. So we use mechanical digestion through the peristaltic action of the large intestine, the squeezing and the pushing. We bring the chyme out from the ileocecal valve, because that's what we still call it, even in the ileum. And then the chemical digestion is the bacterial breakdown of the indigestible material. And of course, the bacteria are very important for us. The bacteria are going to be used to attack the carbohydrates that are not digestible, the cellulose that are found in plants, the plant material, the high fiber material, the bran husk, the fibers in beans, the leafy greens that we like to eat, all that, the cellulose portion of all those structures are going to be attacked by the bacteria, our own bacteria there, and that's what produces gases and acids. A consumption of a lot of fibrous food will often result, because bacteria are doing their thing on the indigestible cellulose, they process it and it's going to be passed out, but it's also going to produce a lot of methane gas as it comes out. And that can often cause a lot of pain and discomfort in the colon. If you have, if you like to eat cooked cabbage and raisin bran for some reason at the same time, that's going to result in a lot of fermentation activities that are going to make a lot of gas and make you very uncomfortable and feel very bloated. And it's going to make you pretty unpopular to the people around you too. We'll let it go at that. So, but of course, we also use the bacteria to make our B complex vitamins, not B12, but all the other B complex and vitamin K. So the
B12, the only one that's not.
Oh, B12, we have to have it, get it in our diet. We have to we have to eat it. So we break that down with the intrinsic factor from our stomach.
That's the only one.
That's the only one, yeah. So, okay. And, okay, so the cecum is, slide's probably out of order here. I already said that. So now the three regions of the colon are the ascending colon, the transverse colon, and the descending colon. The ascending and descending colons are against the back wall of the abdominal cavity. The transverse colon is near the front of the abdominal cavity. So here we see ileocecal valve, cecum, appendix, ascending colon, transverse colon, Tinea coli, descending colon, sigmoid colon, rectum, and anus. Now, what they look like, the ascending colon, nothing special in particular here in its shape. It's designed to receive material from the cecum and move it up towards the transverse colon. Now, the transverse colon is very distinctive in shape. It has a triangular shaped lumen. So it's very easy to recognize because of this triangular shape. Now, the descending colon, again, gives us that spiral structure. You can see straight down into it here. As I was, I'll just repeat this for you. That's okay. The transverse colon is the only one that has that triangular shape. Very distinctive in shape. Now you'll notice that they have what appears to be the pliques also to help move the contents along. Particularly the descending colon, you can almost see the entire spiral ring structure going down in there. These are from colonoscopies, and you will never see a colon that looks this way normally because they're going to have all sorts of stuff inside there, contents. It's only this clean when a person's having a colonoscopy, but this is what they look like. There's your sigmoid colon. The descending colon terminates in the sigmoid, and that leads into the rectum and then the anal canal. So, and the anal canal, the rectum goes through the pelvic floor, and you have the anal canal, then you have the anus. The anus is controlled by two sets of sphincters. like we saw on the urinary bladder, we have a voluntary and an involuntary sphincter. The internal sphincter is involuntary, it is smooth muscle. It will open up long before the external sphincter, which is skeletal muscle, opens up. But it is also under voluntary control to a point. When the pressure inside the anal canal reaches a maximum that the body can tolerate, that external sphincter will open up. So while we do have control, we can't hold it indefinitely. So, and this is what we see here. Here's the internal sphincter here of smooth muscle, and the external sphincter is outside of that, and that is skeletal muscle. Okay, there's the rectum. And now the process of defecation is a parasympathetic response, parasympathetic stimulus. We move fecal matter into the rectum, from peristalsis. The rectum expands, and that expansion of the rectum triggers a reflex, and that is called the spinal defecation reflex. And that triggers the parasympathetic nervous system to tell the sigmoid colon to start contracting, and telling the rectum to start contracting and to relax the internal sphincter. And so we start seeing fecal matter moving into the into the anal canal. The internal sphincter has opened, and now we're putting pressure on the external sphincter, and we have a certain amount of control to open that external sphincter. So it's all about the parasympathetic nervous system, because the parasympathetic system, I always said it's rest, digest, and defecate. So the parasympathetic system controls defecation. Now this is a blockage. This is a blockage in the ascending colon. The dark area here is fecal matter that has been trapped in the colon. You can see it's not a complete blockage. Past this, the descending colon has bits and pieces here. The transverse colon, and it, just to show you, it doesn't go across like it does on the models. It doesn't go straight across. It tends to sort of go up and around like it does here. But we have some sort of obstruction in the ascending colon here, preventing waste material from moving along here. Don't know what caused it, don't know the outcome. This was a student of mine from a couple of years ago, and they were happy to share these pictures with me. So I sort of insisted, but that's beside the point. Anyway, but you can see what happens. You can see what happens here when we get these kind of, we can get an obstruction in the colon itself.
Would that be something to see with constipation from opioids?
Could be, yeah, because opioids are morphine based, and they shut down the motility of the large intestine, and that's what causes the constipation. So that got ruled out, but we don't know what caused this. Never did find out what the cause was. It eventually cleared up. As they said, the problem passed.
But it could also happen from just like low fiber diet, right?
Oh yeah, that's a very, very good possibility.
Maybe some daily thing.
Not enough fiber to keep things going. Even though it's always been argued that a high fiber diet is healthy for you, which it is, but its cancer preventing properties. Well, every year there's a new study that says yes, and then there's another one that says no. Fiber doesn't hurt you, but it doesn't have any significant properties. We don't know, So, because the big study that said it didn't have any effect was done with tens of thousands of health professionals over a 10-year period, And, they knew they were in the study, they knew they were eating fiber, and of course, they're going to eat healthy on everything else. So they skewed the whole results. So what you need to do is track a bunch of average citizens, for 20 years and have some eat bran muffins every day and have some not, and see what happens. So, okay, now, the problem with colons is the possibility of colon cancer. Now, colon cancer, the good news about colon cancer, it's a very slow grower. It takes a long time for colon cancer to get started and grow. Colons normally develop little masses that grow called polyps, and the polyps have a chance of becoming cancerous. A polyp tends to take about 10 years until it gets to about 1 centimeter in size. And after that, then we start paying attention to it. So, the rule of thumb is that your patient that has a colonoscopy generally is, somewhere between 5 and 10 years apart before they have another one, and because it takes these polyps so long to grow that if you have a colonoscopy and you have any polyps and they're taken out, new ones aren't going to be of any concern for 5 or 10 years. So it's when they grow and they get larger that they then become the risk for colon cancer. So, and this is the procedure, the colonoscopy. You place a large tube up through the rectum with a camera and a light source and a cutting device, usually some sort of cauterizing device to cut away tissue and pull it back out. So, and it looks like this under X-ray, but that's not how you track what's going on, because with the camera you can see what's going on. So
Is that hose a hollow hose that's like lit up and hollow on the end, and what they push through the cut with comes out the middle of it?
Yeah, because you don't want to have the sharp cutting tool sticking out. You want to have the light and camera up at the front, obviously. So, but and the it's a fairly large tube. This is why your patients are anesthetized. Now, they used to do these things while your patient was awake. Yeah. But a lot of them, not anymore. That's probably been 30 years since they've done it that way. Yeah, that was, and it's a 30-minute procedure, and then you're going to have to listen to your patients talk about it among themselves for the next 10 years. Get a bunch of old people together and get them talking about their colonoscopies, and they'll be entertained for the rest of the day. So, okay. Normal colon looks good and healthy. Colon cancer, you have a lesion growing there. And even then, colon cancers, while they're dangerous, and then they can kill your patient, the advantage you have is if caught, they're slow growers. And so you can treat it by removing it, and chemo, you can remove the area around there. And if they're caught very early, there's a very, very good chance that they have not metastasized anywhere else. That's the one advantage that colon cancers give you when treating your patient. If they haven't spread, they probably weren't going to. If they haven't spread, then you caught them in time. Okay, now this is what a colonoscopy looks like. This is at least a partial. This is going up in. It looks like it's hard to tell, but it looks like you're in the beginning of the transverse colon there. This is only a short loop of it. But again, you're never going to see it look this clean for real. So, and let's see. I have somewhere, I'll have to find it, a bad colonoscopy. So now, one of the accessory structure that we use is the liver. The liver is our largest gland in the body. It's over here on the right-hand side, right underneath the diaphragm. It has 4 lobes. It's held in place by a ligament called the falciform ligament. The liver makes bile and goes out through, the bile goes to the gallbladder from the hepatic duct. And then we have the cystic duct, I think that's what it's called, taking it, the bile duct, taking it into the pancreas. So we store bile in the gallbladder. We don't make bile there. So there's our gallbladder. There is the cystic duct, which leads from the liver to the gallbladder. We have the bile duct present here. And then here is the sphincter, where the bile duct and the pancreatic duct come together. So it's a very useful organ. It's a commonly removed for lots of people. People that, when the gallbladder becomes inflamed, it's usually because of the presence of stones, gallstones that are formed inside the duct, which are very painful, which can cause nausea and vomiting and fever and pain. Feels like you have the flu. A lot of pain. Can be anywhere here around the right side, up over the shoulder if necessary, for that pain. So now the bile emulsifies the fat. It breaks down the fat. We know the liver also produces heparin. Heparin is our body's natural anticoagulant. The liver produces albumin, which is the number 1 protein in our plasma. Albumin is used to maintain osmotic concentration because it's particles. And we make fibrinogen. Fibrinogen is the precursor of fibrin. The liver detoxifies. The liver stores glucose. It stores glycogen and copper and iron and vitamin A and B and B12 and D and E and K. And the liver activates vitamin D. So the liver does a lot for us. When the liver becomes cancerous, it is a very significant condition. Now, depending on when it's caught, the liver cancer, it can be treatable and your patient can live a long life. Despite fighting the liver cancer. If it has continued to grow, we see this situation here. Notice how big the liver is here. That blue structure on the left is an individual's hand lifting up the liver off the dissecting tray. Now, the average liver is about this big. Let me show you here on the camera. This is the size. This is an average liver right here. I'm holding it in my hand. It's about, let's put it down here. It's about 8 inches across. That's the average size human liver. There's my hand for reference. The liver you see in that picture is about 4 times that. So the liver, those are all tumors on the surface of the liver. Look at the size of the gallbladder underneath there. The gallbladder is about the size of a little kiwi, a little fruit. And it's massive too. So.
Now, is it big because of the cancer or because of obesity that it will grow that large? Or?
It could be any number of things. It could be the bile being produced by the liver. Who knows what the liver is doing with the cancer cells inside there. Okay, bile. Production of bile is the number 1 function of the liver for digestion. Bile is yellowish green. That's why the gallbladder looks green. Bilirubin is the pigment in there. We get that from the breakdown of hemoglobin. Its role is to emulsify fats. We simply chop the fats up into little pieces the same way that dishwashing detergent chops up fat in the sink. we store it in the liver all the time. I mean, in the gallbladder all the time. Bile is stored in the gallbladder all the time. The presence of fat in the duodenum triggers the release of the hormone cholecystokinin. Cholecystokinin triggers the gallbladder to release bile and tells the sphincter, the hepatopancreatic sphincter, there where the gallbladder and the pancreas come together to release the bile and the pancreatic juices into the duodenum. So it is the presence of the cholecystokinin triggered by the, chyme enters the duodenum, that triggers the release of cholecystokinin. The pancreatic duct and the bile duct open up where they're joined, and we release the contents into the duodenum, and we chop up the fat, and we finish breaking down proteins. There's our gallbladder right there. Again, now problems. Gallstones can form in the bladder. The bile can crystallize, and it can crystallize into very large and painful stones. And they can block the bile duct. And so the bile duct can become inflamed. And you have various complications here. You can have chronic gallbladder attacks. You can have acute gallbladder attacks. The inflammation of the gallbladder is called cholecystitis. It may result in surgery to remove the gallbladder because the stones are there. Usually that is the simplest approach to solve the problem. This has been triggered by lots of bile being produced and stored. Now, does your patient maintain a high-fat diet? Do they maintain a low-fat diet? It just depends on whether or not they're going to have a gallbladder attack. If any, Dr. Grice, you've seen Dr. Grice around here. Okay, you know how thin she is. She has continuous problems with her gallbladder. She certainly doesn't fit the profile of what you would expect a person that has gallbladder disease, but she has chronic cholecystitis. I mean, if she looks at barbecue, she gets sick. So I'm not telling any tales out of school. She knows I'd say this about her because that's what happens to her. She readily shares her stories about, gallbladders. So I don't know why she doesn't have it taken out. So that's a whole other story. Anyway, your patient with gallbladder disease with an attack, it hurts. They have a fever, they have pain, an intense pain on the right-hand side. They have nausea, they have vomiting. Any number of things can trigger it, often followed by ingesting something that's high in fat. So, okay. And there we go. Now, I said all that. And okay. And that is the end. Oh, we're at the end. That's good. So, okay. I think I've talked enough about the digestive system anyway. So, and I'd end on that cheery note of gallbladder disease. So,
perfect.
So, anybody have any questions on anything? Okay, then I'm going to call it a day, and we will reconvene next week. And we'll wrap up fluid and electrolytes.